viruses. A lot of what I'm going to tell you about today is not in your book. Viruses were first identified actually as agents that infect bacteria. They were called bacteriophage. And what we're looking at here is an enhanced electron micrograph of bacterial viruses, otherwise known as bacteriophage, recognizing a bacterial cell and attacking the bacterial cell. So what is going on here is the bacterial cell, in this case C. coli. This is a common bacteria that we find in our gut. And the bacterial virus that attacks this particular cell is called T4 bacteriophage. It's fascinating, isn't it? What's happening here is the bacteriophage is actually injecting its DNA into the E. coli cell, and it's going to take over the E. coli cell and use all of the machinery in the E. coli cell to replicate and make more bacteriophage. We'll talk about that in just a few minutes here. Okay, so viruses are fascinating entities. They can come with either a protein coat or a membranous envelope. The bacteriophage T4 that we're looking at here, this is a protein coat. But a lot of the viruses that we have to contend with have just simply a phospholipid bilayer. That's called a membranous coat. It's a phospholipid bilayer that contains the virus. Viruses can have either a DNA genome or an RNA genome, depending on the virus type, and we're going to talk about that today as well. Viruses are not considered living. They're not considered living, even though they can play holy heck with our own cells and make life miserable for us. They're not considered living organisms, and that's because they actually require the cellular machinery to transcribe their genome, to translate their proteins, and also to replicate the genome. So viruses can't exist on their own. In order to replicate and make more of themselves, as what you're looking at here, they need a living cell to infect. A living cell to infect is absolutely critical for the virus to be able to replicate itself. There are many different types of viruses that are known to cause human epidemics and suffering. Measles, mumps. You're really not experiencing measles and mumps because you've been vaccinated against these particular viruses, but nonetheless, for individuals who are not vaccinated against measles and mumps, infection by these viruses causes serious illness. Same with chickenpox. Chickenpox is the virus that causes that. It's called varicella zoster. And what will happen is if you develop the chickenpox, if you're not vaccinated against this, once you have chickenpox, the virus actually lays quietly next to your nerves throughout life. And what will occasionally happen is the virus becomes reactivated and forms a skin problem called shingles, very, very painful. Then, of course, we have the flu. Now, I'm going to spend some time on the flu because the flu has actually been responsible for more deaths worldwide than most other viral epidemics put together. We have smallpox, which another one that we're very happy to be able to leave behind. I'm going to talk about AIDS, or human immunodeficiency virus. And in our next lecture, I'm going to spend a little bit more time on the Ebola virus that causes hemorrhagic fever. Okay. I'm also going to touch a little bit on some of the viruses that are known to cause cancer. Cancer-causing viruses are herpes virus 8, causes Kaposi sarcoma. This is actually a cancer that you primarily find in individuals that have AIDS or are infected by the human immunodeficiency virus. There is the hepatitis B virus. Again, another one that you have blessedly been inoculated against. Hepatitis B virus is associated with the eventual development of liver carcinoma or liver cancer, hepatocellular carcinoma. And then you have the human papillomavirus. Human papillomavirus causes warts, but human papillomavirus strain 16 is a real problem. Human papillomavirus strain 16 causes genital warts, and it is the cause of cervical cancer in women. So for women who are not infected with human papillomavirus strain 16, you're actually pretty good. You don't have to worry. But if you have this infection, what happens is the virus actually will hang out in the cells and can eventually cause cervical carcinoma. Okay. Viruses have also been critical for cellular advances in cell biology. They are a source of all kinds of critical enzymes to be used in biochemical experiments. They're agents for gene therapy. But a lot of experiments with viruses have revealed a lot of critical cell biology pathways that we know now quite a bit about as a result of studies on viruses. So not only are they a major human scourge, but at the same time, we've been able to use viruses to our own advantage, certainly on the scientific front. Okay? Does anybody have a question, or can I repeat anything? Okay. All right. Viruses, like I said, have varying structures. They either will have a phospholipid bilayer. It's called the membranous envelope. Some viruses have what you would call a protein coat. So viruses will have one or the other. A phospholipid bilayer studded with transmembrane proteins. It's called the membranous envelope. And then you have viruses that have a protein coat. What I can tell you right off the bat is viruses that have a protein coat, viruses with a protein coat can actually hang out on a surface. They can hang out on a surface and infect just by hanging out on a surface. You come along, you touch that surface, and you can become infected by the virus. Membranous viruses that are actually within a phospholipid bilayer, these viruses are more fragile. They don't hang out on surfaces very well, and so consequently the best way to contract a membranous virus is to contract it from another infected individual. Okay? And there are a number of examples of membranous viruses. I'll go through that in just a second here. But let's take a look at the different structures of viruses. Plants, not only do we have viruses, animals, birds have viruses, plants have viruses, and like I started out, bacteria have viruses. So there are viruses that specifically will infect all host ranges, plants, animals, as well as bacteria. You're looking here at a tobacco mosaic virus. This matters to you if you're a smoker or if you're a tobacco farmer. This is what they look like. They have a protein coat and they have an RNA genome. Adenoviruses. Adenoviruses are viruses that can infect humans as well as other animals. They have a protein coat that actually forms this wonderful tetrahedral shape. And within the protein coat, you have the bacterial genome. Influenza virus. Influenza virus is a membranous virus. AIDS is also, or human immunodeficiency virus is also a membranous virus. And they have phospholipid bilayers studded with transmembrane proteins. These transmembrane proteins actually allow for the viruses to attach to specific target cells. I'll talk about that shortly. Then we have bacteriophage, and a wonderful example of bacteriophage is bacteriophage T4. T4 has provided huge numbers of enzymes that, for example, I use in my own laboratory. You could not, it's amazing. It looks like a science fiction creature, right? What this thing does is it attaches to its host cell with these little tail fibers. And then what it will do is it will inject its DNA into the target cell through the tail sheath. How about that? Okay. All right. So now what I want to do is I want to describe to you different life cycles of the different types of viruses. And the first type of virus I want to describe to you is the life cycle of a DNA virus. Viruses are categorized by the type of genome that they have, and this would be the life cycle of a DNA virus. And in this case, the DNA virus has a DNA genome. It might have a membranous coat or a protein coat. The virus will attach to its target cell, usually as a result of cell surface proteins. It's the proteins on the surface of the target cell that allow for the virus to actually bind to the target cell and get incorporated in. Once the virus is incorporated into the target cell, the protein coat or the membrane are removed from the genome. The genome is now available and out in the cell. The genome, in the case of a DNA virus, the viral genome is going to have to go into the nucleus of the cell, because in the nucleus of the cell, the viral DNA genome is going to be transcribed by the host RNA polymerase to make messenger RNA. That messenger RNA undergoes modifications such as splicing, 5' 7-methylguanine cap, 3' poly A tail, so that the viral RNA can exit the nucleus and go out into the cytoplasm, where it's going to be translated by the cellular ribosomes. Okay? Also, what happens in the infected cell nucleus is the viral DNA has to be copied. It's going to be copied by the host DNA polymerase to make more viral DNAs. These viral DNAs will also exit the nucleus. They exit the nucleus and they will be incorporated into a new membranous or protein coat, because the proteins that are making up the coat for the virus are called the capsid. These are being translated out in the cytoplasm, so the newly generated viral genomes, DNA genomes, are bound up within the newly translated capsid proteins to form new viral particles. These viral particles can exit from the infected cell, and they're available now to begin infecting the adjacent cells around. Okay? That's the general life cycle of a DNA virus. Again, it's a DNA virus, so its DNA has to go into the host cell genome, so it's going to be transcribed by the host RNA polymerase to make viral messenger RNA, and the viral DNA is going to have to be copied by the host DNA polymerase to make more viral genomes. Again, the viral messenger RNA, the viral genomes are going to have to exit the nucleus, go out into the cytoplasm, where the viral genomes will get incorporated into new viral particles, because the viral proteins that make up the coat are also available in the cytoplasm. Okay? Does anybody have a question, or can I repeat anything? All right. So this is a DNA virus. What's interesting about some DNA viruses is they have the ability to actually integrate into the host cell chromosome. That's what makes it really interesting. And an example of that is another bacteriophage that's been heavily studied, bacteriophage lambda. Bacteriophage lambda revealed that that viruses actually have what is called a lytic and a lysogenic cycle, and I'm going to go over those very carefully as we speak. So what happens with bacteriophage lambda? And you're looking at the structure. This bacteriophage lambda recognizes its host cell. It will bind to the host cell and inject its DNA into the bacterial cell, as you see here. Sometimes the phage DNA will circularize and form its own little genome in the bacterial cell. And what will happen in this case, and this is what is called the lytic phase, the bacterial genome is going to be replicated by the bacterial DNA polymerases. It's going to be transcribed by the bacterial RNA polymerase, make bacterial messenger RNA, and eventually our big viral messenger RNA. The viral messenger RNA is going to use the bacterial ribosomes to generate viral proteins, and eventually what's going to happen is that infected bacterial cell is going to be filled with mature viral particles, with the DNA genome, with the protein coat, and eventually so many viral particles will fill that infected cell. The infected cell will actually lyse and release the mature bacteriophages out into the environment, where the bacteriophage can then recognize another cell and begin the process all over again. This is called the lytic cycle. It's called the lytic cycle because what it does is it fills the infected cell with viral proteins and DNA until enough viral particles are made to cause the cell to explode. Okay? Sometimes, and this is what's not fully understood, sometimes instead of the bacteriophage infecting the cell and going into the lytic cycle, sometimes the bacteriophage will circularize, and then what it's going to do is DNA is going to integrate into the host cell chromosome and form what is called a prophage. Called a prophage, where the viral DNA is integrated into the bacterial chromosome. And the prophage hangs out. It just hangs out in the bacterial chromosome indefinitely. So the bacteriophage DNA is propagated in the chromosome. It's maintained from generation to generation. And what's not understood is what causes this prophage to all of a sudden exit the bacterial chromosome to initiate a lytic cycle. That's not fully understood. It's not fully understood. What is the lever or the switch that causes this prophage to exit the chromosome and function in a lytic cycle? That's not understood, but it can go from prophage to lytic cycle. And when you have a bacteriophage integrated into the host cell chromosome, it's called the lysogenic cycle. Lytic cycle versus a lysogenic cycle. Okay? Can I explain anything else again? Is this relatively clear? Question?
Can you explain the lysogenic cycle?
Okay, the lysogenic cycle occurs when, after the bacteriophage has infected the E. coli, and you have the circular bacteriophage genome in the infected cell, instead of this circular bacteriophage genome generating more bacteriophage genomes and being transcribed into bacteriophage proteins to make mature phage particles, instead that genome integrates into the bacterial chromosome, and it's now called a prophage. And it just hangs out there indefinitely until a switch is flipped and the prophage can actually emerge from the chromosome, go back into the lytic cycle. Okay? And again, it's not understood. It's not yet understood. What is this switch? This is important because we have viruses that do very much the same thing. Okay? All right. So let's take a look at some of the major scourges of humankind and what kind of genomes they have and what they cause. So we have adenovirus. Adenovirus, it has a protein coat, not an envelope, but a protein coat, causes respiratory disease. They tell you it causes tumors, but that's under very rare circumstances and not in humans, as far as we know. Then you have papavirus. Papavirus are the famous papillomaviruses. Human papillomavirus would be an example of this. And human papillomavirus causes warts and cervical cancer. It also has a protein coat. Then we have the herpes viruses. Herpes virus, simplex 1 and simplex 2, so that's cold sores and genital sores. You can find simplex 1 on the genitalia. You can find simplex 2 on the lips, depending on just how you have your fun. Use your imagination, okay? All right, not a guarantee. Okay. Then we have chickenpox virus, Epstein-Barr virus. Epstein-Barr virus is a virus that causes mononucleosis, and also is associated with the development of a cancer called Burkitt's lymphoma. We've all been exposed to Epstein-Barr virus. Sitting in this room, we've all been exposed to this virus, but most of us will manage to get by without developing mono. And Burkitt's lymphoma is a tumor associated with Epstein-Barr virus, but it only happens in Central Africa, where the patients who are exposed to Epstein-Barr virus, which is endemic in the population, have also been exposed to malaria. So it seems to be an overtaxing of the immune system. Then we have pox virus. That's smallpox virus. And thank heavens we don't have to worry about that anymore. It used to be very, very debilitating and scarring. Another wonderful virus is the reovirus. Reovirus, example, is rotavirus. Believe it or not, rotavirus kills more children in the world, in the developing world, than almost anything else. It's a intestinal virus, causes diarrhea to the point where the infant will become dehydrated and die. So many, many children in the developing world will die as a result of rotavirus infection. Okay? How would we treat rotavirus infection in the United States? Pardon me? Well, that doesn't work so well because the child is vomiting furiously. IV. You treat them with an IV, keep them hydrated, just keep them hydrated, and eventually the immune system will clear the virus and the child will recover. But in the developing world, they don't have that luxury. Okay? So rotavirus is a serious virus in the developing world, kills children. Then we have everybody's favorite, the coronavirus, the common cold virus. We all enjoy that from time to time. Flavivirus. I'm going to talk about this in our next lecture. Flavivirus